So, transcription and translation is known as protein synthesis. Transcription is when DNA transcribes into RNA, it copies itself by an enzyme called RNA polymerase. Enzymes use one strand of DNA in a gene as a template to make messenger RNA. The RNA polymerase and transcription factors bind to the promoter. So there are 3 codons at the very beginning, and that's what it binds to. This causes the DNA strands to pull apart. One strand is the template that makes more messenger RNA, and the other strand is the sense strand. RNA polymerase starts the mRNA development at the start of the codon and then moves to the gene in the elongation process. So there's the 3 codons. The codon is the 3 bases at the front. Then the mRNA is made from the 5' end, and RNA nucleotides are attached to the 3' end. RNA synthesis is a process of creating RNA molecules. RNA polymerase makes RNA molecules from the DNA sequence. Gene regulation is a method of determining which genes will be turned off and which will be turned on. And that's learned through the proteins because they have a certain job that they do. And that's how they determine where they're going to go in your body and in the cells. Proteins bind to certain gene regions to increase the rate of transcription. Proteins can also decrease transcription and even stop it. RNA polymerase puts the DNA strands back together after transcription has occurred, so only 10 to 20 bases are ever exposed. So once it's opened, then it zips closed. RNA polymerase 1, 2, and 3. Type 1 is for ribosomal RNA, and it's found in the nucleolus. Type 2 is the messenger RNA, and that's found in the nucleoplasm. And it's also inhibited by the alpha amanitin. And then type 3 is the transfer RNA, and that's found in the nucleoplasm, and that makes the 5S protein and the rRNA. So that's the ribosomal RNA. Introns are the DNA that makes up the genes, but they fall out because they just get recycled. They don't do anything for the proteins. And then the exons are the DNA that make up the gene section of the mRNA, and that's the one that becomes translated into the protein. It can also get added to other ones, so then we have longer proteins. How cells prepare RNA to leave the nucleus? Well, the mature mRNA has to be only exons, so all the introns have to be released, and then the proteins, because of what the codons read, whatever's transcribed in there, it tells the proteins what their job is. RNA editing, so that's the coding of the genes. The messages change. The mRNA can add, delete, or delete the nucleotides. C can be converted to U, cytosine to uridine. The MECP2 gene can have a mutation, and the ADAR cell will insert, delete, or convert nucleotides. So it sounds like our cells are, or the RNA, the transcription is trying to prevent mutations. Then there's translation, which happens after transcription. That's when the messenger RNA is coded for a specific protein. So that's the codons, the 3 codons. There's 3 bases in a codon, and they read what that protein is supposed to do and where it's supposed to go in the body. It inhibits or it promotes whatever its job is. It also gets carried by the tRNA, which is the transfer RNA, and it's also linked to amino acids. The arrangement of the nucleotides in the codons is called the reading frame. This codes for all the amino acids. Each codon corresponds to a particular amino acid. AUG is the start codon. It's the promoter and the initiator. It's going to start the translation. Then we have UAA, UAG, and UGA, which are all stop codons, and they're terminators, so they stop the translation. All of this happens in ribosomes. There are ribosome subunits that connect to mRNA and an initiator, the tRNA that attaches to the start codon. So the initiator is the tRNA, and that attaches to the start codon. Then larger ribosome subunits join to finish the translation initiation complex. The tRNA that corresponds to the next codon after that start codon will join the ribosomes with an amino acid. The first tRNA detaches and leaves the ribosomes, making room for the next tRNA, allowing a new amino acid to join. This process just keeps on going, and that's how polypeptides are formed, and that's how they make a protein. The DNA is transcribed into mRNA, and that translates into a protein. The structure of ribosomes, they are organelles, and they help the cell to make proteins that it needs to function. The location of ribosomes in the cell will determine the protein it makes. They are made of RNA and protein and are made in the nucleolus of the nucleus and then released in certain spots to do their jobs, to make the proteins to do their jobs. There is a large subunit and a small subunit derived of ribosomal RNA and ribosomal proteins. The subunits stay separated until the ribosomes need to make protein production, and then they sandwich over the RNA, and that's where all the coding takes place. So a protein are the genes that have the instructions for making proteins. RNA polymerase attaches to the gene strand. The strand splits, and the RNA polymerase makes a mRNA strand out of the bases in the nucleus. The DNA code determines the order of the bases in the mRNA. So there's the three bases in the codon, and then whatever those are, that's where the RNA is. Those are the other letters, so A to T, C to G, and then RNA has U. Ribosomes bind the messenger RNA, and they help produce the chain of amino acids. The tRNA carries the amino acids to the ribosome. So the amino acids all attach together, and that's where they create the elongation of polypeptide chains. That's how the peptide chains grow. So all the bases in the codons have to go through the ribosomes. Then the amino acid is released, and then the 3D shape of protein is made. So the polypeptide is the chain of amino acids linked together by peptide bonds. Amino acids are the sequence of nucleotides in the DNA. So that's adenine, thymine, guanine, and cytosine. Monomers are building blocks of proteins. The structure of primary, secondary, and tertiary, and quaternary. So primary structure is the sequence of amino acids that make up a protein held together by peptide bonds. So that's just the straight peptide chain. And the secondary structure is the amino acids beginning to fold either in an alpha helix or a beta pleated sheet. And the amino acids are arranged in such a way and connected by hydrogen bonds. And the hydrogen bonds determine if it will be a helix or this pleated sheet. Then the tertiary structure, this is the folding of the 3D shape. So that's when the peptide chain wads up into a shape and it has its job. And then it goes and does its job. What determines the shape are water loving and water fearing amino acids. I don't remember which one. One of them, I think that the water loving, so hydrophilic, those are on the outside, and hydrophobic is on the inside. Then there's the quaternary structure, and that's when one or more of the polypeptide chains come together. So there are some mutations that can occur. The mutations are a change in the genetic material. Some mutations are silent and they are never known. Sometimes the bases aren't matched correctly, and that's called substitution. Sometimes a base is added, and that's called insertion. And sometimes a base is removed, and that's called deletion. Mutations are random, although some factors can increase the probability of mutation. There are external factors like chemicals and radiation. There are also internal factors like that happen in the replication and the interphase phases of DNA, replicating the bases and the codons. The codons read the bases. I guess the codons are the three bases. Sometimes that can get mixed up. Genes code for proteins that influence traits. So one or more bases has to change, and that's going to produce different proteins making different traits. The proteins have a job, and the code of the DNA tells the protein what it's going to do. When the bases get either added or removed or mixed up, coded wrong, it tells the protein what to do, but it's not right. Extra copies of genes can be made. When the genes get deleted, they just break off. There's inversion, which means that the broken chromosomes get reversed and reapplied to the chromosome. And there's also translocation, which means that the broken chromosome attaches to another chromosome. There's frame shift. That means when the order of the protein synthesis is mixed up, like it's inserted or deleted. There's also inversion, which means reversed. So the bases in the codon can become reversed, so they break off and they can attach back and they can be reversed. They can be mixed up. Single nucleotide. So the only thing I got with that was single nucleotide polymorphism. And this is a gene mutation for single base pairs. So bases can be left out, they can be added or substituted. These all cause genetic differences, but not all genetic differences are bad. They create differences in all of us, makes us all unique. Sometimes the mutations can change the protein shape. The proteins can either be long or short, and depending on how long or short they are and what the codons are telling the protein to do, it could make it spiral or the pleated sheet. So how to read the codon map? Also, with the hydrophobic and hydrophilic elements in it, I wonder if that could change too. I don't know. So how to read the codon map? So there's two of the codon maps. There's a rectangular and there's circular. So the rectangular is you would read the first letter from on the left, and that will give you the row, or maybe the row. And then you go up to the top and read the second letter, and that's going to give you the column. And you come over to the right and you read the third letter, and that's going to give you the exact row. So we've got the row, the column, and then the specific line, I guess. And that's going to tell you what that codon is doing, what that codon does. Then with the circular one, you read it from the center out, the same process, whatever that is. With then you find another A in that section or whatever the next letter is, and then the last letter. It just breaks it down into smaller sections. RNA moves to different parts of the cell because of the protein functions. The proteins tell the RNA where to go and what it's going to do. Mechanisms that will change proteins during transcription, depending on environmental conditions. So, gene regulation and expression are regulatory proteins, also transcription factors, that attach to promoter and enhancers that tell the protein what to do. Operons are regulated clusters of genes along with promoters and an operator. A protein called eukaryotic initiation factor 2 helps translation begin. It can be regulated, and sometimes a phosphate group is added to it. So it helps kickstart the transcription process. Let's see. How the systems we create can affect someone's protein synthesis. So things around us can affect how our bodies transcribe and translate proteins. If we like the drugs that we take, um, our diet, our exercise, our hormone balance, which is kind of a big thing because there's studies showing that, you know, how women take birth control and then we pee in the toilet and then that water gets recycled and cleaned, but you can't take the hormones out of it. So we end up ingesting that. Men ingest it, and it's kind of messing things up. I don't know. That can affect things. Um, the amino acids needed to build proteins can be inhibited. Um, Sometimes the chemicals we put in our bodies can stop or accelerate the wrong proteins to affect the production within our cells, like testosterone in men. Um, are we responsible for someone's phenotypic expression, including behavior, by creating systems and environments that affect protein synthesis? I think this depends on the situation. Obviously, I'm not doing anything right now to affect anybody, but a pregnant woman can harm her fetus and change the phenotype. Sometimes she doesn't have to do anything, and it's just little happy accidents that happen sometimes. Um, I think that there is quite a bit of evidence to provide the proof that we need that the environment we live in does affect our bodies to the cellular level, and it can affect our DNA. Um, I think a good example is Chernobyl. Um, Ultimately, the radiation uh, didn't have to be put in a big container, but we did that. But it did explode, and then people were damaged. And then for centuries later, generations later, uh, they're still dealing with the effects of the radiation uh, from their parents. So I would say that we are responsible for someone else's phenotype depending on the situation. Obviously, that's not black and white. But I think a lot of the issues that we face are because of the environment we're in, the food that we eat. I think that we're all connected cellularly, so um, how our bodies process things and the different chemicals in our bodies can really affect um, how DNA can be transcribed and how the different cells, how the mRNA transfer or moves the proteins throughout our bodies.